To split human embryonic stem cells grown on Matrigel to Matrigel, we start with a confluent 6-well plate of embryonic stem cells on Matrigel, which can be split 1:3 to 1:5 to another Matrigel plate. This allows the wells to become confluent again 4 to 5 days after splitting. On the day of splitting, wash each well that will be split with 1x PBS pH 7.4. Then add 1 milliliter of 1 milligram per milliliter disc paste to each well and incubate at 37 degrees Celsius for 3 minutes. Wash each well 3 times with 1x PBS pH 7.4. Then add 1 milliliter of embryonic stem cell media without BFGF and use a cell scraper to scrape the colonies off the bottom of the well. Collect the resuspended embryonic stem cells in a 15 milliliter Falcon tube and pellet by centrifugation at 200 times G for 5 minutes at room temperature. To replate the human embryonic stem cells on Matrigel plates, first wash the Matrigel plates with DMEM F12 and add 2.5 milliliters of MEF conditioned media supplemented with 10 nanograms per ml BFGF per well. After the cells have been centrifuged, resuspend the human embryonic stem cell pellet in an appropriate volume of MEF conditioned media supplemented with 10 nanograms per ml BFGF. Pipette the cell suspension up and down 3 times. Keep in mind that the stem cell clumps from Matrigel break up easily, so pipetting 3 times should be enough to break the clumps but not make the colonies too small. Finally, aliquot 0.5 milliliters of human embryonic stem cell suspension per well to achieve 3 mils per well as a final volume. Make sure that the clumps of cells are evenly dispersed and then place the plate in a 37-degree incubator. Depending on the particular embryonic stem cell line, embryonic stem cells will become confluent again 4 to 5 days after splitting, when colonies will start to touch each other. It's important to know whether or not your human embryonic stem cells maintain pluripotency or stemness in culture. To check pluripotency of the cells during embryonic stem cell culturing, we use immunofluorescence microscopy to examine the expression of the pluripotency markers OCT4 and SSEA4. One of the wells in our 6-well plate is fixed and stained for OCT4 and SSEA4. If we see that the cells are fluorescently labeled for both of these markers, then we know that our human embryonic stem cells have maintained pluripotency and can be used for our experiments.
We've just shown you how to grow and maintain pluripotent human embryonic stem cells on mouse embryonic fibroblast feeder cells and under feeder-free conditions on Matrigel.
So that's it. Thanks for watching and good luck with your experiments.